Good morning everyone. This is Trishna Ball. I am an assistant professor at Department of Pharmaceutical Sciences and Technology, BIT Mesra, Ranchi. The second part of this lecture mainly related to the preparation methods of polymeric nanoparticles for targeted therapy. The most common methods that are utilized for preparation of polymeric nanoparticles are emulsification and solvent evaporation or extraction process. The second method is nanoprecipitation method. The third method is salting out method, and the fourth method is supercritical anti-solvent method. Now, as you can see in the very first method, that is the emulsification and solvent evaporation or extraction method, this method basically depends upon the phase separation conservation technique. As you can see, the first phase, it contains of the organic phase, as well as there is the second phase, that is the aqueous phase. The organic phase basically it contains the polymer which is dissolved in an organic solvent. There are various categories of organic solvent like chloroform, dichloromethane, ethyl acetate. These are the common organic solvents that are used for the preparation of this organic phase. Various types of synthetic as well as semi-synthetic or natural polymers, if it is soluble in these organic solvents, they are utilized and then they are dissolved in the organic solvents, and that constitute basically the aqueous or the organic phase. Now, this method, it has an advantage that this method is only utilized for this lipophilic drug. In one sense, we can say that this is a disadvantage or limitation for this method. It cannot be utilized for the hydrophilic drugs. So, here you can add this lipophilic drug directly into this organic phase, or you can disperse this lipophilic drug into the aqueous phase. Now, coming to this aqueous phase basically it contains double distilled water or millipore water or distilled water, as well as it contains a stabilizer in the form of a surfactant, so that the formed nanoparticles, they do not adhere to themselves. Now, when this aqueous organic phase is added dropwise to this aqueous phase, there should be a continuous stirring of this aqueous phase for the solvent evaporation, so that the phase separation conservation can easily take place. Now, the disadvantage or the limitation of this method is it is high energy requirement for this method, that is high energy requirement utilized for this method for the homogenization, and this is the main hindrance which is basically seen in this method. Now, this stirring for this whole process should be taken at room temperature. There should not be any hiking in the temperature. Depending upon the stirring only, the solvent evaporation of the whole procedure will take place. The second method, that is the supercritical anti-solvent method, this basically contains an anti-solvent, that is carbon dioxide it is used. Now, the solute containing the solution is sprayed in the form of tiny droplets into the high pressure vessel containing liquid anti-solvent, that is carbon dioxide it is used. The rapid diffusion of this carbon dioxide into the solute containing the solution results in the formation of nanoparticles. The main advantage of this technique, it includes an omission of organic solvent. As you can see in the previous method, we have seen that there is utilization of organic solvent, and for the complete removal of organic solvent, there should be a very high speed homogenization is to be utilized, and also care is to be taken that the residual solvent should not be left behind in a precipitated form on the surface of the nanoparticles. So, here in this method there is no utilization of organic solvents. So, there is no such tendency of such kind of organic solvent being precipitated on the surface of nanoparticulate systems. The therapeutic proteins are very well encapsulated into these PNPs by this method. So, this is the biggest advantage of this method, that very like therapeutic proteins which can get precipitated in presence of the organic solvent. Now, this method can be utilized for the encapsulation of these therapeutic proteins, large DNAs, macromolecular proteins that can be easily encapsulated into these polymeric nanoparticles via this method. The third method, that is the salting out method, this basically it contains here it is an emulsification process. Here the emulsification of an organic phase, that is the polymer solution in acetone, as we have seen in the last case, into the aqueous phase takes place first, and salting out is achieved by adding a high concentration of salt, that is electrolytes like magnesium chloride, calcium chloride and magnesium acetate, or sucrose, which is a non-electrolyte, to the formed emulsion. Now this method, and once the emulsion is formed, uh, like uh, then it is being stirred or homogenized at a high speed, and there again the precipitation or the phase separation co-observation takes place. Now this method avoids the use of high speed shearing, surfactant and chlorinated solvents. In presence of this salt only, the precipitation takes place or the salting out takes place. Other advantages include high drug encapsulation efficiency as well as easy scale up procedures. However, extensive washing of the nanoparticles is mandatory in this technique because there is a possibility of retaining of these salts that we have mentioned, that is magnesium chloride, calcium chloride or magnesium acetate onto the surface of these particles, of these nanoparticles. The fourth method, that is the nanoprecipitation method. Here as you can see, the first polymer is dissolved in a water miscible solvent, that is acetone, ethanol, hexane and methylene chloride, any of these solvents, and then it is added drop wise into the aqueous phase which contains a stabilizer or a surfactant, and once it is added, then the phase separation co-observation of the substance takes place and the result particles that we get in the form of nanoparticles. The advantage of this method is high drug encapsulation efficiency, narrow size distribution. There is no need of high homogenization and easy scale up is main advantage of this method, and disadvantage is only lipophilic drug can be encapsulated into this PNP by a nanoprecipitation. Now I shall give you some more information regarding this nanoprecipitation method, that is organic phase which contains the polymer and the water miscible organic solvent. It is added drop wise into an aqueous phase which contains the stabilizer and water. Now this organic phase which is basically a water miscible solvent, and the polymer is basically water immiscible. So as it comes in contact, the water miscible solvent gets miscible with the aqueous phase, and when the water goes more into the aqueous phase, the polymer now co-observates out from this mixture of the water miscible solvent, organic solvent as well as the aqueous phase, and thus you get the nanoparticles. Now this, as you can see, I have already told earlier also, the stirring should be at room temperature and no high speed stirring is essential in this method. In case of salting out method, organic phase which contains a polymer dissolved in water miscible organic solvent, same as that in case of nanoprecipitation method. Drop wise addition of this organic phase into the aqueous phase takes place, which contains the stabilizer, water and the saturated electrolyte or the non-electrolyte, and the high addition of high quantity of water. Now this when you add huge amount of water into this, elimination of the solvent takes place and the salting out agent by the cross flow filtration takes place. Now this salting out method can be taken place by this method also, or it can also be carried out by addition of high quantity of your salt, as I have mentioned that use of various salts like magnesium chloride, calcium chloride as well as magnesium acetate. Now these are the nanoparticles which are being synthesized by any of the above mentioned procedures, are finally collected by ultracentrifugation or high speed centrifugation, and they are washed with millipore water or millicu water 2 or 3 times, lyophilized in presence of cryoprotectants and stored at low temperature until use. Use of cryoprotectants makes them stable, free flowing, and thereby also helps them from prevention of various shrinkage, from letting them not lose their structure. Now the various polymers that are used for preparation, as I have already told you earlier some of the name of the polymers. Now these are some of other polymers that are widely used, like polyvinyl alcohol, polyethylene glycol, monomethoxy polyethylene glycol MPEG, polysorbate, vitamin E. These are the example of hydrophilic non-toxic biocompatible polymers that are used for surface modification of these PNPs. Now these polymers provide a cloud of hydrophilic chain onto an outer surface of nanoparticles to prevent the process of phagocytosis and opsonization by circulating the plasma proteins in the blood. Thus, this surface modified amphiphilic drug delivery systems containing an anti-cancer agent within its hydrophobic core surrounded by a hydrophilic outer shell ultimately have increased systemic circulation time and persistence in the blood. Additionally, this PNP based targeted drug delivery system boosts the tissue specific distribution of the anti-cancer substance via an active and passive tumor targeting without affecting the healthy cells. Now, this is an example of how the passive targeting, this part is the passive targeting, and here we go for the active targeting where you attach a specific ligand, and here there is no specificity. So, active targeting is much more advantageous than in comparison to the passive targeting. During the passive tumor targeting, this PNP easily reach the tumor cell due to the defective leaky structure of the tumor cells and impaired lymphatic system. We have already discussed about the enhanced permeation retention effect, whereas during active tumor targeting, a ternary structure enters the tumor cells or tumor vasculature via receptor mediated endocytosis. This ternary structure is composed of a ligand as a targeting moiety, a polymer as a carrier, and an active chemotherapeutic agent. Now, this ligand is a specific ligand that are used to functionalize the drug loaded PNPs. The different type of ligands that are used to decorate these NPs, that is the nanoparticles, include small molecules, peptides, proteins, antibodies, engineered antibody fragments, and aptamers. Specific receptors, which are like membrane bound domains expressed on cancer cells or on the tumor endothelial cells, are the main targets for these ligands. The folate receptors, the epidermal growth factor receptors, as well as the transferrin receptors are mostly present on the cancer cells. On the other hand, the integrins, vascular cell adhesion molecule 1, aminopeptidase, membrane type 1, matrix metalloproteinase, and vascular endothelial growth factor receptors are expressed on the vasculature of tumor cells. The other outstanding features of this system include non-toxicity, marvelous biocompatibility, and bypassing the resistance associated with the multiple drug dosing. The polymers that are tested for designing of NPs based on targeted drug delivery system with and the most outstanding candidates among all those are the polyhydroxyalkanoids, poly and cyclodextrins, and polylactic glycolide, which are synthetic, natural, synthetic, and synthetic. Besides these, various types of natural polysaccharides are also being utilized for the preparation of these nanoparticles. In the next class, we will be discussing about some of the other methods that are utilized for preparation of nanoparticles. Thank you.